out of 38 years in groundnut. I thought this title will be more suitable for this presentation. Okay, thank you, Natraj. I think we can move on to the next slide. And we have here two selection procedures with hybridation and without hybridation. Before actually the variability was exhausted, in the beginning, initial stages of the crop, when this was recently introduced, actually groundnut is a recently introduced crop, around 300 years old, through different trade routes into our country. It is originated in South America, Bolivia, and the Andes Mountains, where naturally this groundnut is grown in the wild form. It is seen there, and it is recently introduced in terms of it is not a crop of our own country. It is introduced crop. So in the beginning, whatever that varieties developed were through these two methods, mass selection and pure line selection, and varieties developed. Next slide, please. Move on to the. This mass selection will actually will be like looking similar looking wood plants, and we'll be collecting the parts from those plants, and we'll be pooling, and the resultant bulk, if it is pure line selection, will be growing plant to progenies, and best progenies only will be selecting, advancing as a variety. Ultimately, all the yield trials will be there. They will be evaluated, all these selections. Ultimately, we end up with a variety. Next slide. And these are the varieties that were developed in groundnut through mass and pure line selections. TMV2, that is selection from Gudiatham bunch. Mass, it is a mass selection. Here, good-looking plants, similar-looking plants. Part that through in terms of plant as well as part characters, they were pooled, and this variety has come into existence in 1940. One more variety, it is a selection from germplasm line, JL24. This has also become popular in 1980. 80s. It is the variety that ruled during 1980s. And it is a selection from germplasm line EC94943 from Jalgan Center. Like that, we have the mass and pure line selections we call JSB11, AK1224, local red or Polachi red. Actually, one red natal was introduced through different one seed, one trade route. Mostly these are all through sea only at that time, through ships. And this red natal, the selection from that might have been named as Polachi red in Tamil Nadu and adjoining states of Andhra Pradesh. And in Virginia, M13, Kadri 1, Kadri 3. Kadri 3 is a selection from Rabot 33-1. Kadri 3, it has served as a base for many, later on also, for many varieties. Maklu red in Virginia runner bunch types. These are in different states. These were the varieties selected through simple these mass and pure line selection procedures earlier. Next slide, please. And as we know, this variability it got exhausted in all these introductions and land races. Later on, only way to create variability is through hybridization. To some extent, mutation breeding also it is possible. But in groundnut, through hybridization is the major source of creation of variability. Next slide, please. And before we think of hybridization, we have to choose our parents. This choosing of parents, of course, these are all dealt in detail by Dr. Nikam and earlier speakers also. I just wanted to stress again here. Objectives have to prioritize before selection of parents. What for we are going to breed, and what for this hybridization is. And we may have different constraints, but major constraint only you have to prioritize first. We have to take into consideration. And of course, genetic resources, if the constraint to tackle that constraint, we do have, whether we have the genetic resource, that is that trait under consideration in a genotype, in a germplasm resource, or in any advanced breeding, we have to think of the genetic resources available before choosing our parents. If there is information and combining ability of the parents also, we have to search literature, and we have to know the combining ability of the parents that we are going to choose. that will help in a long way in getting goods and this nicking ability. If it is good, we are definitely we'll be getting good segregants in the next segregating generations. So if it is available, it is always better to consider combining ability also of the parents that we are going to choose. We have to search literature and we have to take if there is that information, we have to take that information also. Generally, we use popular high yielding varieties as we want to rectify the defects in these varieties as female parents. And if the resistant parents are any trait that is of interest is in a genetic resource, if it is reported, it has to be again verified its resistance or the behavior of the trait. It has to be verified again at our place. It may not behave in the same way in our place. So it is very much necessary that we have to verify it again in our place if it is behaving the same manner or not. Otherwise, just entering, taking that from literature and directly, you should not go for hybridization with that line. And we have now this magic, that is multi-parent advanced generation intercrosses. And Dr. Nigam was also telling about, discussed about this in detail. We can restrict our crosses up to 3-way crosses only. Up to that, we can easily identify F1s and we can judge whether they are in the segregating generations also, whether they are segregants or not. So that will be in our purview if we go for this crossing program. Up to 3-way, we have to restrict up to 3-way crosses only. We should not go beyond that. Then otherwise, it becomes very difficult to identify whether it is a really segregant or not. All these things will crop in. So it is better to go. Maybe there are some other reasons why we are going for magic, but for development of a variety, we can better restrict up to 3-way crosses only. Next slide. And again, this number of crosses that we have to be affected. And if it is a major constraint, we may have to simultaneously improve 3-4 popular our varieties, like this Narayani, Tirupati 4, and Kadiri 6, like that. We may have to improve all this for that trait under consideration. Then we may have to go for more number of crosses. Again, number of donor parents available also. That is also a constraint. May not be always the case that we have many donors. So donors for maybe for resistance to late particles, like that, we may have many donors, but for some of the traits, stem rot, we are not finding that many donors. Of course, now recently some interspecific derivatives have come and we have sufficient number of parents there also. And we have to consider our resources, land and crossing block available also before deciding the number of crosses that have to be affected. Actually, this is the advantage of conventional program. We can tackle actually more than 2-3 traits together in a crossing program. It is not only 2-3 traits, maybe more traits also. We can together, that is integrative selection we can make through conventional breeding. In that way, we have to think of our parents also and crosses also. And in farming, in making crosses, we can decide on some matting design. We can derive some information also in that process, and we can have some papers also published. That is also important for youngsters. And in the same they can be in line into tester or DLL. This information and GCA, SCA, it will go in a long way in deciding the actually combining ability. This is very important, and baking ability. When we have very good general combining ability, parents with high GCA, we are sure of getting improvement with respect to that trait. In that way, we can this information will help in deciding which crosses we have to handle in which way. Selection procedures also we can decide based on the GCA and SCA of the parents and SCA of hybrids. Next slide, please. And this is an example I am taking this. These are the hybridization stands that we use for making crosses in Tirupati center. And if it is not always the case that we have to choose all these high yielding existing varieties only, Here we have a case like this ICGV 06188 and 07359, 07368. These are bold seeded with high pod yield and with low shelling out ton. Here our objective is improvement of shelling out ton. Then we can use our high yielding varieties. Dharani, it is a very high sheller. We get up to 80% shelling out ton with Dharani, even 1073 also. And during Rabi, we get around 76 to 78 shelling, 78% shelling. As we want to improve shelling out ton in these female parents, these are our high yielding varieties are used as male parents in this program. Actually, the success rate again it depends in humidity and weather. Normally, we go for hybridization in Tirupati during Rabi season, unless there is a it is a student program, unless if it is a part of our any collaborative program. Otherwise, we go for crossing program during Rabi season, as we have cool weather and high humidity. Success rate will be very high. And even in Tirupati itself, we have achieved the success rate of 36 to 40% here in Rabi season. But in ICAR setup for under controlled conditions, they're getting around more than 80% success rate in in crossing program. Thank you. Next slide, please. And this is dial set of crosses. Here in dial set, it is half dial set. Just to acquaint you what is dial set, what is a tester and learning to tester, and I have just kept this list of crosses. Here we'll be getting all crosses in dial fashion, all combinations. Some in line into tester only female parents and male parents. Here there is some overlap also will be there. Like Tirupati one Narayani, we won't that is not a cross that we want. But in this, when we attempt crosses in dial pattern, some crosses like this also crop in. But some material, if it is good, then we can take, and otherwise we can reject it in the F2. But we can get combinations like GPPT4 and ICG-FDR 79 here in this dial set crosses. That is the advantage. Some useless 2-3 crosses, if they are useless, Narayani, Kadri, they're very closely related parents. Narayani, like that, Tirupati 1, Kadri 6. They may not be that desirable crosses in the pattern we get, but some information will be getting through this dial set of making through this dial set of crosses. Next slide, please. This is line into tester here. Tirupati 4 is female parent. All the this is having this is crossed with all the testers here. ICG all the 6 testers. Like that, we have with each parent we have all the crosses with the testers. Like that is the advantage with line into tester pattern. Mostly breeders, they go for line into tester design, as we want to improve our existing varieties with the addition of traits from testers. Okay. Mostly testers are our genetic resources or donors, we can say. Next slide, please. And coming to F1 generation, this is the most crucial generation we have to take care of, like our just bonds. This is a very important generation where the recombination takes place, and we'll be getting more number of recombinants if we grow them, take care and grow them in an ideal situation. And confirmation hybrid in the field is very important. If it is not a hybrid, generally we assume that F1s are uniform. But if there are many selves, if there is if adequate care is not taken care of, adequate care is not taken at the time of making crosses, we may end up with selves also sometimes. And it is very actually crucial. We have to identify F1s in the field at different stages. We have to observe F1s and identify the F1s. Even at the time of harvest, we have to look into again keenly. We should not harvest as in the yield trials. This one, we have to take care, and each plant we have to see and whether it is F1, if it is F1, it is okay. Otherwise, if there is some doubt, we just keep it away and raise it separately. If they're actually, we consider F1s to be informed, but usually in the field we see many selves intermittently. So it is always better to individually observe each and every plant during the crop growth period as well as at the time of harvest. And one more thing I just forgot while choosing, I wanted to share actually at the time of choice of explaining about choice of parents. When you are picking up the material for making crosses, you do it yourself. Don't delegate this work. Material, picking up the material. Maybe Tirupati one, we are going to pick up, you're asking someone, maybe sub-assistant or technical assistant. He may pick up about maybe Tirupati one label, but exactly that may not be the same. If there is some mislabeling, whole program will collapse. I advise you to just pick up the material by yourself for making crosses. That is very much necessary. That if there is some mistake at this stage, I think the whole program, it will be a futile exercise. Just do it by yourself. Scientist, you have to do it by yourself, picking up the material with the label, and you will be acquainted with the material also sometimes. If there is some mislabeling, there will be some problem. If, for example, if I ask you to get up and get tag 24, if you are new to that to the craft, you will depend on only label. Sometimes these labels, they will be misleading. No, I should not blame the breeders, although sometimes it happens. So you should be very careful while selecting the material and see that material twice and get that material at the time of sowing, stand at the hybridization stands and do it get it done under your supervision. Okay, next slide, please. Next slide, please. One. And just I wanted to show these are F1. Some cells will be there in the in between. Just that the red one is again self. These blue ones there, which ring in the blue ring, they are hybrids. Like that, we have within a row we have to see and identify the F1s. These are the parents. We can side by side we can grow the parents. Again here also labeling. And at the time of harvest, there should not be any overlap. Okay, you have to be very careful while harvesting. You have to stand there. Okay, next slide. And these are some of the F1s and parents. The middle one is F1. Okay, both the parents, just a depiction. Next slide. Next slide. Move on to next slide. Identification confirmation, it is already I have explained, but if parents are more diverse, identification F1s is very easy. If they are very close, I think it is very difficult even for experienced people. And generally, we won't advise to make crosses between closely related or derived from the same cross advanced breeding lines. Unless it is very necessary, why there is a small hitch, then you can think of like that with the development of nils or reels like that. But it is always better to choose parents which are more diverse. Of course, depending on the objective, if they are more diverse means again some linkage drag, other things also will crop in. Depending on the objective, you have to choose the parents and identify the F1s. If actually if they're taking into consideration morphological traits and again flowering pattern, each canopy structure, everything you have to observe in the field and then identify the F1s. If we cannot identify the F1s between closely related parents, what we can do is we can grow separately each assuming that each is F1 and plant to progeny rows we can raise in F2. And if there is segregation for the characters that we are targeting, then we can select in those progeny line, that F1 progeny lines only in F2. Otherwise, reject. Next slide. And in handling segregating populations, that is first segregating population is F2. This is the pedigree method is the most popular method. And this is of where we can see the recombination, segregation, all this, the effects of all this that have happened in F1 through meiosis. They will be all visible in your first segregated generation F2. F2 population size, as I said, it should be large as possible, but it should not be too large. Also, we can't carry on, or we can't I select in such a large population. Also, it becomes a stupendous task for the breeder. Optimum size will be around 500, so that we can capture most of the segregants, extreme segregants. That way, we can. I have to target our F1s then we have to decide the number of parents that we have to sow as female parents as well as male parents. In that way, we have to plan that crossing program so that at least 500 plants will be able to get in F2. F2 500 means at least 50 to 50 F1s should be there. The F1 is handled properly. Each F1 plant around 30 parts. Like that, we have to calculate and decide the number of female parents that we have to sow for making grasses. And again, this number of plants that we have to select individually in individual grasses, there is no restriction. In some grasses, actually, we'll be seeing lot of variation because of maybe the nicking ability of the parents involved. We see lot of variation. We select as many plants as possible in such grasses. In some grasses, there won't be any variability seen. It may be due to many reasons that I can't explain here. Maybe that hybridation that at that time we could not make coincidence of this male and female flowering might not have happened. That in that grass, maybe that is why we might have done less number of pollinations. That might be one of the reasons. Otherwise, the nicking ability itself is may be low between the parents involved if the variation is low in a particular class. So if there is more variability, you can select more number of plants. If less, like only few plants we can select. Otherwise, we can reject all the whole grass as it is in F2 itself. Next slide, please. And F3, we see family characters. Just some uniformity will be seen. And within progenies, there will be some variation still, but between progeny variation will be more, within progeny variation will be less. We can reject here any of the inferior ones, mostly. And if there is more uniformity, you can at this stage also, we can select whole progeny, but this should be in very less number, unless maybe the trait that we are considering, we have to think. Next slide. Next slide. In F4, I think mostly the family characters, they will be very clear and more uniform. The progenies, we can, as a whole, we can select in F4. And if there are one or two plants that are varying, we can just reject them. And some progenies, we can select now. And we can, if the seed material is sufficient, we can just go for yield trials with those uniform bulk selections. Next slide. This I have shown just to say that by F4, F5, uniform bulks will be more and single plants will be less. And all the uniform bulks, if they are more uniform and if sufficient seed material is there, they can enter into replicated row. They will be, can be promoted to replicated row yield trials. Next slide. Next slide, please. And in F5, similar manner, we will be selecting superior progenies. And while we are promoting to yield trials, consider acceptable market rates along with yield. Just don't promote based on only pod yield. You look into pod and seed traits, particularly acceptability. Without these acceptable pod and seed traits, if we are promoting, it will again end up with a variety that has high yield potential, but it is quality wise, it is very poor, and acceptability by the farmers and by the consumers, it will be, it won't be there. And if it is only for oil purpose, even then Our main aim is oil also should be good, no? It should be peltable. That way, if you are attempting some distant crosses, in that you please think of this acceptable pardon seed rates. Many breeders, of course, they're not that much serious with respect to pardon seed rates. Please consider without sound mature kernel percentage, rose testa is the preferred rate, red testa in some regions. Mark shelling out, and it should not be lower than 70 at least. It should be more than 70 at least. I think now these year people also have taken much care in promotion of entries. But what about the entries that are already promoted and in pipeline? And so, in some cases, I think there is some, what is that, mismatch there, where the entries are not getting promoted because of this the higher pod yield of entry in this varieties with poor spot and seed rates. We are not, we don't know what will happen to the entries that are there in pipeline with superior pod and yield rates, and they won't be promoted. That has to be looked into by our DGR people also. And superior progenies harvested en masses, they should be promoted to replicated trials in, they will be mostly uniform. Next slide. Next slide. These are more uniform progenies in F5 generation. This is from cross TCS 1157, TCS 1043. We will see here that compact nature of TCS 1157 and ashy coat with ashy coat. Many progenies are with ashy coat. From that is Dharani. Next slide. And these are again just to show how these UVs, they will be maximum in F5 and minimum these SPs will be lower. And from here, they will be entering into replicated row yield trials. Next slide, please. This is replicated row yield trial. It is not necessary that we should go for replicated row yield trial only. If you have enough material, you can directly, if you are confident of the material, you can just put them into initial yield trial also. Initial yield trial and evaluate and then promote to advanced varietal trial. If that is that liberty, I think you can have a replicated trial again. If there is sufficient seed material is not there, then you go for replicated row yield trial. Then take caution, each row will be sowing, and at the time of harvest, you have to be very careful here also. Next slide. And these are some of the recombinants I just wanted to show with the ashy coat in this three-way cross, tag 24 TCS 1157, TCS 1043. Next slide. Next slide. In AVT stage, we show our material to our administrators also here and our some fellow interdisciplinary to have interaction and to know the acceptability and other traits. We show to our ADR is there, Dr. Giridhar Krishna. One agronomist is also there, Krishna Reddy, and we'll be discussing whether how are these advanced breeding lines, which one is better. Of course, with their interaction will be fine tuning again in promotion. Next slide, please. To next stage. This is the material that we are drying Kharif produce. Our university head just came in and he's looking that material. I'm just explaining. We use this kind of trays here in Tirupati and plastic bags also, netted bags for drying. Next slide, please. And whole exercise, the making crosses, all that, this success of the whole hybridization program, it will depend on all these things, emasculation, whether pollination are done with adequate care, and stage of the bud, at which I think that they will be at the base of the plant. You should be very carefully select those buds, and without much damage to the stigma, that pollen should be removed. And we can take help. We can't do everything by ourselves. And for emasculation, pollination, we can take skilled person's help. But while under our supervision, this has to be done in the evening and in the morning. Pollination, emasculation in the evening, pollination in the morning. We have to be there personally to supervise them. True F1s are identified, and these segregating generations are handled with interest. I am saying that you should have that passion. In F2, if we see that segregation, and in F3, if you find some good segregants, and you should have that excitement that your breeder should feel. That is possible only with pedigree method of handling segregating generations. And other methods of handling segregating generations won't allow you to see that, to visualize that segregation pattern, and that excitement won't be there. You can see at each and every, from F1, F2, F3, you will be with the material in pedigree method. I feel that pedigree method is the most, but you should have that interest, that passion with the material, and then only you will enjoy that. You have to stand in the sun, of course, for long hours at the time of harvesting, and after, at the time of drying also, you have to take care. At each step, you have to, there should be proper follow-up, and there should be again in the after harvesting, drying, you have to check the material. And here it is not exactly pedigree record, pedigree breeding at like that. You can have a list of selections in each cross at your in your record, and then verify then after drying, and then whether you see whether that material is there or not, and after drying again check all the seed and part rates, then only promote to next generation. Rejection is also very important in each and every step. Otherwise, we'll be having a lot of material lying. You can't handle them properly. You just reject them if they are not proper. At every generation, that rejection also it should be done ruthlessly. That is, then only you will be successful. And in each class, there is no hard and fast rule that this many number of selections only has to be brought into field trials. But if a class is good, you can have some 20 selections also, not more than 20, then otherwise it becomes very unwieldy. And in some classes, there won't be any progenies or any selection that is promoted to field trials. Okay, now inferior material anyway will be rejecting in F2 and F3 generations. That has to be done. There is no need of promoting inferior classes, selection from inferior classes. We can right away throw them in F2 and F3 itself. Next slide, please. We can have some indication about the performance of the cross through evaluation of means, evaluation of yield in early generations. You can study these genetic parameters also. If there is any MSc student or allotted to you, you can get these things done, and it will serve our purpose so that these cross combinations only can be handled with high mean variance and heritability. Next slide. Next slide. And these are some of the genetic gene actions responsible, which confuse the breeders: penetrance and expressivity and threshold. And these confuse the breeders in the selection process. Penetrance, first of all, whether the character is expressed or not, expressivity, that level of expression. And there will be for some characters, there will be thresholds also. And modifying factors also, epistatis, linkage, they interfere with our selection process. I know these are known things. I need not have to go into details of epistatis. What is epistatis, pleiotropy, and linkage? Linkage and pleiotropy should not become confused. Influence of one gene over or many genes is pleiotropy. Linkage is due to the presence of the genes on the same chromosome. Okay, next slide, please. It interferes with the recombination process. And we have, for some of the characters, we can't visualize them. With the eye judgment, we can't visualize them. Usually, we can't select. For such characters, we need markers for selection. And we have very good equipments now. This pad chlorophyll meter reading, that is actually it is a surrogate trait for drought resistance. It has proven by our physiologists. And this has become a very handy tool in the hands of breeders to check the drought resistance capacity. And like this, we have this for high oleic trait. Again, we have good equipments, tools for assessing the high oleic acid content. These selections we have to rapidly feed them and get the values of fatty acid levels in those selection in the recombinants. And we should employ markers also. We have some allele specific markers for traits like this for selection in initial generations itself. We can select using the markers, allele specific markers for high oleic trait we have. Like that, for other traits which we can't visually judge, we can go for markers. Next slide, please. And this bulk population method, this is known, but it is not that much useful for groundnut breeders. I feel we have to, every time we have to harvest in bulk and sow a similar part. There is no any control here over the variation, just usual one bulk we will be harvesting F2 generation, similar plot from the harvested bulk will be raising. In the last for F4 or F5, we'll be selecting single plants in the material. Next slide, please. Next slide. It has only one advantage here. In initial stages, these epistatic and dominant effects, they will confuse our selection. They won't be there in advanced generations. It is not followed frequently by our breeders because of the plot size. And as I said, pedigree method, we have that scope of applying expertise and skill in selection that we can't do in early generations and through in bulk method. It is a long time process again. Next slide, please. A single seed descent. Here we select each one part from each plant in F2 generation, and in the next generation we raise it from these seeds. Like this, population almost remains constant. And this of F5 in F5 or F6 will be selecting single plants as in pedigree method. Here almost all the variation in the initial generation, that is from F2 it is maintained up to the end. That is the only advantage. But skill and expertise of the breeder is not we can't apply in early generations in this one also. That is the disadvantage with this method also. Next slide. And as this is useful for development of recombinant inbred lines for markers work. Next slide. This method, backcross method, it is useful only when we have to rectify one defect in a popularly grown variety. For example, Kadri 6 susceptible to foliar disease, we want to introduce late leaf spot and rust. Then it is okay. Otherwise, it is employed to rectify one defect or one or two defects. And here also making back crosses, that it is not that easy. Instead of that, we carefully do pedigree method itself we can do. And every time back crossing and the yield level, there won't be an increase. We can have that defect rectified. If it is threatening the existence of very resistance of the crop, that kind of problems only we can go for this method. And here again, the number of plants of recurrent parents should be more large enough to have BC1 F1 at least 200 plants. Next slide. Next slide, please. Next slide. We move on. And this already I explained, through backcross method of breeding, oleic acid content was improved. F435, it is the mutant, natural mutant that has high oil content of 80%. This was used in developing Sun Oleic 95R. Through high oleic breeding program, we were also collaborators with ICAR and DGR for this program. In that process, ICGV 15083 and 15090 were identified for release as GIRNAR. I think they were released as GIRNAR 4 and 5 from DGR. Next slide. And this is the robust tool that we can use for the assessment of fatty acid levels in groundnut near infrared spectroscope. Next slide. And now we have come to the end of that first part, where variety development, what are the different steps that we are going to follow in after hybridization up to the variety development process. In F5, we will be ending our material in yield trials We'll be promoting therefrom. We will be organizing replicated trials, initial varietal trial, advanced varietal trial. The material that was found that is found promising in advanced varietal trial, it will be again promoted to MLT at state level. And if it is promising, then it is promoted to mini kits. This is the procedure that is followed for release of varieties in at state level. At the central level, that at AVT stage, the material is multiplied and given to varietal test, the ASRP testing. And then there also again from initial varietal trial, if it is promoted to AVT, and then if it is identified at AVT stage, it will be become a variety. And this my journey, this I want to just briefly just follow just my journey in agro with groundnut at RIRS Tirupati. I was at Tirupati almost in RIRS Tirupati almost 30 years. So the promotions, all the things happened during my stay at RIRS Tirupati only. Next slide, please. Next slide. I joined as research assistant there. When I joined as a research assistant in July 1986, at that time, our earlier breeders, our predecessors, I have to say, they are actually very keen in a collection of the material, and they have collected and evaluated germplasm lines from ICAR-IIGR and other places, and they have they had some two trials at comparative varietal stage, that is advanced varietal stage. And they at that time the target is the replacement of TMV2 and JL24. That is their target in Spanish group. In Virginia group, they wanted alternatives to local Pedigutti, Kadri1 and Kadri3. In two trials, the material was actually almost 10 to 15 genotypes only in each trial. And one ASAP trial was also there. These are the only trials at the time of my joining. But next slide, please. I have actually I had that stupendous task actually of handling 30 to 40 crosses material. They had material for earliness, drought resistance, yield, foliar disease resistance, all in from F4 to F7, lot of material. And they have sown in almost 3 to 3 acres. And myself and one more a senior breeder, we actually made selections at the time of harvest. Here, the donors for earliness were EC 21137-1, AH316S. These are all Valencias maturing in 95 to 100 days. These were used as donors. One more EC 21083A. This is for yield. For FDRS, these are the 5 lines that they have used, PA 393527B. These FDRS lines, they were well in EC 76446 and NCSC 17090, NCSC 17133F. These are all Valencias. These two, PA 393527B, these are mostly towards Virginia. And this formed my MSc material also, FDRS material. These 5 lines were used, and that material with the Kadri1 as the female parent, that material was given to me in F2 generation at that time. So it is grounded by the time I joined. It is not a new to me, and it is not a new crop, as I had done my MSc in groundnut only. Next slide, please. This we brought all this material into yield trials in IUTs, and we had 8 trials together in 88 Kharif, and around 200 selections we have evaluated. At that time, our people, they have good bulbs, actually identified as good bulbs from JL24 AH316S. The smooth parts red testa. AH316S is a red testa line. JL24, it is susceptible to mid-season stress. That is why in the in-between it has, once it was popular, only in the early 1980s. After that, because of this drawback, it faded away actually from the cultivation. That is why we have, otherwise it is a very good variety. So our people have taken that and improvement of that. In most of the crosses, they have used TMA2 JL24 only. And from that material, actually, in comparative trial, these were TCZ 1704, 1706 were identified as high yielding lines. From Virginia bunch, PCC 1518. From 2 trials, these are the promising materials that are identified. In the initial stages in IET trial also, some good promising ones, we have included them in hybridization program. That is the stage. We don't have any material, but any material that has come through ASRP, it has served as our germplasm at that time. Next slide, please. And as I said, this is the 1704. It is a selection from germplasm line EC 106983/3. And just why I am explaining all this is, you can learn from here how the breeding program has progressed from initial stage to later. Now the full, it is in full form at RRS Tirupati. And it is this line is a kind compared to TME2 we have selected. It is released in 1989 as Tirupati 1. Actually, it is a very good variety at that time, but because of the seed production, lack of seed production in large scale, we could not actually that variety has not become popular, I have to say. Otherwise, it is a very good variety. Still, we use this line as one of the parents for crossing program, and this is selected as a reference variety also. Okay, next slide. Next slide, please. This is Tirupati 1. Parts are almost of similar size to the TMV2, but shelling out it has very high shelling out of almost 79% also. Attractive testicular is also very attractive. It has ashy leaf coat. At that time itself, they were very keen and they have selected that kind of genotype, our predecessors. Leaflets are also narrow. In TMV2 and JL24, they are very broad, but here in this one, they are very narrow. Next slide, please. Next slide. Next slide. And this from those bulks which are selected and evaluated in different yield trials, these are the 2 lines that were promoted to next stage, that is release. Tirupati 4 was released first, as it has rose testa. Preference is for rose testa. In 1995 itself. In 2002, Narayani was released. It is a red testa line. But the Tirupati 4 again, it suffered like JL24 because of it doesn't had that. It is actually that stress tolerance at the end of season. Stress tolerance was it is a problem in Tirupati 4. That is why it was withdrawn again. But otherwise, under ideal conditions, it is also a good variety. But Narayani, it performed well even under stress situation. It is an early variety and 100 days variety, and it is through drought avoidance mechanism, because of the earliness, it has become very popular. And it has high shelling out turn. Next slide, please. Next slide, please. This has an early vigor also, which is very important trait for success of a variety in drought situation. It is puts up fast vegetative growth, and that is why it has found a place in rain-fed situation for a long time. Still, it is grown in some of the areas. In Chittoor and Chittoor, Annamaya and Nellore Kadapa districts is the most preferred variety. Red testa, it has preference in some of the areas also. This is Tirupati 4 variety, again attractive testa color, and only because of lack of that drought tolerance, it has not become that popular. Next slide. Next slide. Narayani, this I have already explained. Next slide, please. This is Narayani. You see the fast growth in the photo. That is a uniform early vigor. That is the advantage of Narayani with Narayani. Next slide. And Tirupati 2, this is again a selection from one of the this material, breeding material supplied from ICAR-IIGR, GOG1 and Cifla 14. Initial stages, ICAR-IIGR has given some segregating material also to 2 different centers. This is selection from that material. This has Kalahasti mildew tolerance also. Wood variety for Rabi situation. This one is. Next slide, please. Next slide. And we have a location-specific problem called Hasti melody. This is caused by soil-borne nematode, Tylencorhynchus brevilineatus. And in 1980s, it became a severe problem in Rabi season in Chittoor and Nellore districts along river belts of Penna and Swarnamukhi, mostly in sandy soils. This happens if this is the problem in the soil. If there is a high infestation of these nematodes, these pods become small, thin shell, and the surface also becomes dark, and consequently yield reduction will be there. It affects developing pegs and pods. At that time itself, they have pooled genotypes from ICAR-IIGR and other breeding centers, and they have screened it at the location, the hotspot location, and they have identified this TCG for 1518 only as the resistant one, and was TCG 1706 was found to be moderately resistant. That's out of 1500 genotypes. They are screened in the hotspot location, the field itself where that problem is there. Next slide. This is Tirupati. This is later on released as Tirupati 3. This has high oil. Actually, it is a selection from TMV10, variegated testa variety with high oil content. Our breeders, I have to say, they were very keen in selecting. They have identified a red testa line out of the variegated testa line. Next slide. And this was used as one of the donors to develop short duration variety. This TCG 1518 is a Virginia bunch line maturing in 125 to 130 days. But the problem, this Hasti melody, it is a Rabi problem. Rabi, we want short duration, short staged varieties. And in the hybridation taken up, 15 crosses, taken this as donor 1518. And we have ended up with good varieties there also. Kalahasti, one of the varieties, TCS 320. It is high oil, short staged, and short duration is 105 to 110 days variety. One more variety released. This one was released. Kalahasti was released in 2002. Prasuna in 2006. Prasuna has again attractive color, testa color, and parts are also very highly acceptable. But here this 320, it had, because of its high oil and red testa, it didn't find place. Prasuna and these two are restricted. Almost they didn't become that popular actually because of maybe the pod and seed traits that actually that are there in these varieties. And one more thing, it is just 320. It is more like a Virginia line, though it is a Spanish bunch with respect to this main axis bearing all that. Otherwise, it looks like more number of primaries, more towards Virginia. This happens in infra-specific crosses that we are all actually working now. We are making crosses between Spanish bunch and Virginia runner types only now. That clear distinction also has gone now. And this, though we say that Minax bearing this is Spanish bunch, clearly for a practical purpose, when we have to consider duration only, I feel. How early they are maturing, whether they are coming in 100 days or whether they are maturing in 110 or 125 days. That must form the criteria now onwards, I feel, to decide which one is this Spanish bunch or clear this classification. I think that this has become blurred now because of the infra-specific process that we are making now to transfer traits. That is one more submission that I wanted to clarify here in while actually promotion of entries in IASRP or in our state programs also, these kind of doubts they arise. Next slide, please. Next slide, please. One more in between, we have an insectance breeding program. This I have taken up actually immediately after coming my PhD from IARA. And these are all the donors are from ICAR-IIGR. Though we did not end up with a variety, we got a very good bridge lines, I have to say, 645, 647, 876, 653. And one more, TCS 635 identified with a pentafolate one. It was registered for that also. TCS 653, it has gone up to many kits, but could not promote further. Maybe some problem with other traits, emergence and other traits. We could not promote further. But, before that, I think many good lines from other programs also they have come. And these served as very good donors again for water use efficiency and drought tolerance. Whatever the lines here we have identified, 645, 647, and they have become very good donors subsequently in our water use efficiency breeding program also. Next slide, please. And we handled, actually, I was a collaborator with these two projects. I joined late, but our physiologists were there from the beginning, and here they have standardized all the screening techniques for water use efficiency and drought tolerance. And they have identified in the first program some of the donors also, which were actually next slide, please. And in this project, they have from the experience gained and from the parents that they have identified in the earlier program, they have assigned some common crosses and some location crosses for the 4 centers at the 4 centers: Sikrasad, Junagadh, Jalagan, and Tirupati centers. These are the common crosses, and these are the location specific classes. From our place, location specific classes given 34 tag 24. All these from all these 4 centers, they have pooled the advanced breeding lines 192, and they have evaluated. Next slide, please. Next slide. And here, they have formed they constituted a selection index for selection in trade-based approach, considering harvest index, transpiration, and transpiration efficiency. And here there was gain, no doubt, with respect to harvest index compared to T and TE. But there was no difference among the selections made through trait and empirical in the approaches. Empirical is yield evaluation under drought situation. That is what we follow as breeders. Top 10 genotypes, they didn't differ much with respect to these two approaches. Top 10 in trait or empirical, they're almost on par. From here, from this program, from location specific cross, we came up with a selection TPT 25 with high short stature, high water use efficiency, high yielding ability, and was released as Abhaya. Next slide, please. Abhaya, this is Abhaya. This says all the good traits, high yield potential up to 5 tons per hectare. Also, we could actually harvest during Rabi season. Only small drawback is short stature. When we have sown this in Kharif situation, when there was severe droughts on dry spell in initial stage of crop growth, it has not put up any vegetative growth. And consequently, in the end, the yield harvested was not that much in the Kharif situation. But Rabi, it is I think there is no variety still to beat this one. Okay, this okay. And otherwise, it is a very good variety. Even now, I have to say, very high yield potential. Small drawbacks, they will be there. It has no bee cutter. While shelling, there is some difficulty again here. That difficulty also farmers have expressed. In spite of that, many farmers, they are still interested to grow this variety during Rabi season. That is the yield potential of this variety. Next slide, please. Next slide. This is about Abhaya. I have already explained you this high yielding ability and all that. Next slide, please. Next slide. And because through the material that we got from that more efficient breeding of drought resistant peanuts of this ICAR collaborative project, and we had a lot of actually material, the donor sources, but to fine tune, to narrow down them, and we have evaluated all this 192, and we have pulled some of the lines from different centers also. In the different breeding centers, around 287 germplasm lines we have evaluated. Through this experience, we have proposed a project that to the AP Netherlands Biotechnology Program. At that time, Dr. Emurao was the chairman and Dr. Pakki Reddy was the executive director. we could get this project, and physiologist was the principal investigator, Dr. P.V. Reddy, and we got 55 lakhs for the project. And in the initial stage, to narrow down and to identify the sources of sources for drought tolerance to include a breeding program, we have evaluated again all these pooled lines, and we have identified some top 50. Next slide, please. And from that, we have narrowed down to these 5 lines for inclusion in breeding program as donors for these drought tolerance traits. These are all the drought tolerance surrogate traits, we have to say, with high correlation with the drought resistance. This was verified using the real-time that experiments also, and these were actually crossed in line into test fashion in one set and dial set also among different advanced breeding lines, promising lines among the top 50. One more program is also attempted, but I am confining to this program only. These are all female parents, are all our varieties. VRI2, I have to stress here, this is from Rudachalam. We got a very good line involving this VRI2. Next slide, please. And here I have to stress for 34 cross combinations, TCGP6, it was used both as male as well as female. It is a high water resistant line with acceptable pod and seed traits also. Only again defect is bold seed, and that SMK was not that good. Shelling out and also was not that high in that line. And in total for 34 crosses, we have made 35,846 pollinations. Out of that, we realized 1435 FN plants. FN plants we could realize. In total, 34 cross combinations we have studied. In each class, we had almost 42 FNs. This is a case study we can think of. And the segregating material was handled by modified pedigree method. As I said, F2 population ranged from 500 to 1500. And by Kharif the 2007, the material was in F5 stage. We have good inform bulbs, 133 inform bulbs. We evaluated them in 4 sets in initial varietal trial, and we narrowed them to 41 entries, and they were evaluated in advanced varietal trial in Kharif 2008 and Kharif 2009 also. Next slide, please. Next slide, please. Actually, some fast tracking was done to meet the project objectives of AP Netherlands Biotechnology Program, and we entered into yield trial of these, some of the lines, some promising entries from F5 itself. We have multiplied and we have put them in advanced varietal trial, and then gone for on-farm testing. Out of that, we have released this Grishma and Rohini. These are all short-staged varieties with high water efficiency. Next slide, please. Next slide. And I will be taking more time to explain about TCS 1043. This is the most acceptable, high-yielding, drought-tolerant resistant culture that I have seen during my tenure, I have to say. And this, as I said, we have evaluated from Kharif 2007 initial varietal trial, later on Kharif 2008 advanced varietal trial, that for 2 years advanced varietal trial. And we have classified the seasons. Kharif 2007 was very favorable season. Kharif 2008 was not that favorable. And luckily we got that kind of, naturally we got that kind of situation. And Kharif 2009, it was the worst situation. Next slide, please. And I need not have to go into this rain. In Kharif 2007, was very favorable. We got 691 mm of rainfall. There was no excess rainfall, no rain, no dry spell, and average temperature, everything was very optimum, and sunshine hours, everything was very optimum, and very high yield was recorded during Kharif 2007 by almost all the genotypes. Next slide, please. Next slide. It was not that favorable. It was the total rainfall, it was 370 mm only. There was deficit rainfall of 17.5%. There was intermittent dry spells. Crop experienced moisture stress at dwindling phase in this season. Next slide. Kharif 2009, it was very highly unfavorable season. Total rainfall received was only 274 mm from July to October. And though there was drizzles in between, but there was stress at each and every stage: seedling, flowering, pegging, pod building stages. The total at the end, after at the end of the crop season, 183 mm rainfall received, that contributed for fresh germination, again in situ germination, and it didn't help the crop. Next slide. Next slide. In this situation, this is the line. Kharif 2009 was this TCS 1043 was identified with high yield potential, with high shelling outturn of 71%. Even in that situation, that bad situation, with 100 part weight of 81. Next slide. How we can utilize this, the information that we get naturally, that we had, that is I wanted to impress upon you here. See the yield data: 2007 2562, Kharif 2008 at 1066, in 2009 1180 kg for 8 kg per hectare. And this is the advantage that we have recognized during Kharif 2009 with TCS 1043. That was, next slide please. Next slide. And this is, of course, next, please. Next slide. Next slide. This is Dharani, released TCS 1043. This is derived from VRA2, the TCGP6. This has very high shelling outturn, 80% also we can get in Rabi. Next slide, please. And it has actually, see the crop, it makes this primaries also, they make a little bit wider angle. Maybe it is a good light capturing mechanism also, it is there in this, and they were avoiding mutual shading, all that. Next slide. Next slide. And these are the parts of Dharani, acceptable. Next slide. Slightly bigger part in Rabi. Next slide. Next slide, please. Okay, this I say is the best combination. That is a selection from VRA2 TCP6. It has higher yield potential, 17 to 33% higher yield. This optimum SCMR and SLA, all these root rates, shelling outturn, underseed weight, they're all within range. That makes it a more suitable culture for rain-fed situation. It has tolerance to drought, low light, and stem and dry root rots. To certain extent, it can tolerate PB and PSND. It has crop duration of 105 to 110 days. Oil content of 49%. It is suitable for all. Early Kharif, to some extent, it has some disadvantage. It will be putting on more vegetable, but otherwise it is okay. Next slide, please. Next slide. One more combination that is, of course, almost on par with the Dharani, but it doesn't have that much drought tolerance capacity. It is more suitable for ideal situations, Kharif or Rabi. Next slide, please. Next slide. This is TCS 1073, released as Dheeraj, with this has also all the good traits. Once in our place, we could not irrigate during Rabi season, and there was, of course, there was no rainfall at all during Rabi season. And this situation, only Dharani and Dheeraj, they stood in the field. All other cultures, they washed away. They were vitiated. Only these two remained. Next slide, please. Next slide. One more culture that has come out of that breeding program is Pragati. It was recently released in 2019-20, I think. And it is for Zone 3B, Rabi season. Highly accepted pod and seed traits, short statured variety with all the desirable attributes. Next slide, please. Next. One more, actually, one more variety that is popular in rain-fed, in Kharif irrigated situation, early Kharif, and Rabi also is tag 24 in our place. They want replacement to tag 24 because it is highly susceptible to stem rot, and it has that in situ germination problem also, and some other traits like LLS also, it is not at all tolerant, and there will be leaf fall, filling will get affected. To have alternative, we have actually made crosses with the tag 24. In that process, we didn't exactly get that alternative, but this one, this Jyoti, it is a three-podded one grown for wet parts. It is boiled parts. It is actually used for table purpose. Jyoti, this Nityaharita, it served as a good alternative to Jyoti. Jyoti takes time almost 125 to 130 days, Virginia line. This Nityaharita, it is more, though it appears, looks like Virginia, but it behaves like a Spanish bunch and matures in 110 to 115 days with the cluster bearing. Very good pod filling, not like Jyoti. This is a very good alternative, very high yielder also, both for Kharif and Rabi season. This can be cultivated short staged. It is more now picking up in some areas near Visakhapatnam and some in zone 3A also, that is Gwalior region. This is picking up. It is a very good variety again. Next slide. And here, why I am showing this one is three-way crosses. Why we have to go for three-way crosses? Here we have to TCS 1157 1043. Why we have made this? For improvement of shelling out and peck strength. Like that, in between we will be having these traits which we have to add to the varieties. They will be actually, we have to look into those traits, how we have to rectify the recently released ones or advanced breeding lines, and we have to make crosses. And this will be a continuous process, three-way crosses to bring in again the Trost testa and the desirable attributes of these three varieties, three Kadri 6, JL24 and TAG 24 into the segregants into this, bringing into single genotype. Next slide, please. And this is FTRS program. One more PhD student was given. These are the lines that he has used and he has made a dial set of crosses. Next slide, please. Next slide. This is dial set of crosses that he has made. Next slide. Next slide. From that material, PhD material of Dr. Vishnuvardhan, one of the students whom I guided, this sister TCS 1694 was released. This is from Kadri 6 FDRS ICGA 79 cross. Now this is becoming more popular. It has all the, it is highly resistant to late leaf spot and moderately resistant to rust. It has high water efficiency. This now it is becoming popular in Kharif, Rabi, and early Kharif also. It is not that tall also, not that tall, moderately tall. So that is why it is finding place everywhere. 100 seed weight also, it is around 51 grams with oil content of 49%. This is the now ruling variety in many places in Andhra Pradesh, both in rainfed as well as Rabi situation. Next slide, please. Next slide. You can see the resistance leaves, they are remaining green even at the time of harvest. This is our ADR. TCM ICAR-IIGR has visited our plant at the time of harvest. Next slide. Next slide, please. One more that has come out of that breeding material is this Konar TCGS 1707, that is recently released variety. It is from GPBD4 and ICGFDRS 79. Both are resistant parents to late leaf spot. It has a tolerance to sucking insects also to some extent, and stem rot also, besides LLS and rust. Next slide. Next slide. And major program now thrust is on the breeding for this to breed varieties alternate to TAG 24. That is why we have taken up now in recent years this program. Next slide, please. Some of the I have explained mostly where I have directly participated. Our breeders, Dr. John and Dr. Srividya, Dr. Kiran Kumar Reddy, and others have they have their own set of made a set of crosses, and that material is also there. From John's material, this is 1416. It has come out well, but we could not release it because of the pod and seed features are not that good. We have kept it, but otherwise it is a very good high yielding variety. Next slide, please. Next slide. This was released now recently as Himani. It has high this attractive color rose testa, almost light rose testa, I have to say. Next slide, please. Next. This is just I am sharing. So we have actually many advanced breeding lanes. Dr. Siddiq, when I was presenting my RKY program, he pointed out, why can't you pull and study the diversity as well as this drought tolerance, what is that, mapping work from this one. That way we have pulled them. Would not exactly do mapping, but we have pulled them around 350 lines for this work. This is that line. This is that photo of those lines grown. Next slide. Next slide. This is high yield project material in lattice design. We have what we have done. Next slide. It's in collaboration with ICAR-IIGR we have done it. I have to thank Dr. Nigam. We were there as collaborators in isoform seed system project, FV, this farmers participatory varietal selection program, high volic program. All these programs we were collaborators, and we are largely benefited from those actually projects. And because of that, the ICAR programs which we have handled, we got this APNL Netherlands Biotech project also to our center. Okay, this is Backgrass program that we have attempted for development of high volic groundnuts in our center. Next slide. Next slide. In Rabi, that this gap is there now. Many people, they want varieties with confectionery quality. With that objective, we are making crosses. John is looking after this work. Meanwhile, we have a stopgap arrangement. We have, next slide, please. We have identified a line from the material that is supplied from bark. We requested them to supply some bold-seeded lines. Out of the 10-15 lines that they have sent, we have picked up this line. TG47, and we have released at state level. This is a 3-seeded line, short-staged, high-yielding line, maturing in 110 to 115 days. That is the beauty of this one. All the bold-seeded varieties from Kadri or from ICAR-IIGR, they take almost 125 days, but this is a short-staged as well as short duration line in bold-seeded lines, among bold-seeded ones. Next slide, please. And we had, this is one just a part of that. We organized many FLDs, many kits we have organized with on farmers fields. Next slide, just to show that. And many field days we have organized, mostly for Dharani, where I have organized many field days in Anantapuram, Chittoor, Kadapa, many places, even in North Coastal districts, Srikakulam also. Next slide, please. This is the TCS 1073 field day in Chittoor district in Satyavedu area. Next slide. And many plots, many seed village programs, seed production plots. Seed production we have done vigorously actually from 2002. Farmers participatory mode, we have done seed production, large quantities. Now we are almost confident of popularizing our own varieties with our seed production program. Once we release a variety, that there our job won't end for breeders. We have to popularize them. We have to see that the old varieties are replaced, and the benefit of new varieties reaches the farmers, and they benefit from that high yield and our other traits also. Next, that is happening now. And we have very good collaboration with the seed production agencies, Department of Agriculture. That is needed for breeders. We have to maintain good collaboration with the Department of Agriculture, people, farmers, good seed production agencies. Okay, next slide, please. This is our visit of our monitoring team. Dr. Daboria is seen here. Our ADR is there. Next slide, please. Our hybridation stands is our experimental plots. Dr. Daboria and his team. And our team is there, whole groundnut breeding group. Next slide. And these are all the varieties that release from Tirupati Center, starting from Tirupati 1. I started my career when these were in yield trials, Tirupati 1 and 2, Tirupati 3. And from Tirupati 4, we have participated in evaluation and selection also. Okay, next slide, please. This is our seed production strength from 2019. This is I am giving this from 2019-20 only. You see the quantity of breeder seed, foundation seed that our center is producing of different varieties. We have demand for almost all the varieties that we have released now. These are the major popular varieties. Now we have 5 varieties. Next slide, please. Next slide, please. Next slide. This is again from in a different year. This is in brief all the achievements that I have made. I have to acknowledge the help and guidance that I got from our predecessors, Dr. Siraj, starting from Dr. Siraja Reddy, Dr. Ramlingeswara, Dr. Jairam Chandra Reddy, Dr. N. Rajagopal, our earlier breeders, later on physiologists, Dr. N. P.V. Reddy garu. We have very good collaboration with physiologists. That is how we could breed drought-resistant varieties now. We are hopeful of screening techniques, these screening tools that they have developed also. We are used extensively, they are being used now in screening for drought resistance in our trials now, in our breeding programs as well. And I have to thank all our students also, 3 PhD students I have got a major advisor and 17 MSCs. All actually they have worked day and night again, and they have handed over me the material, good material that they have generated in proper form. Otherwise, I could not have handled them. So they have lost that material. That I have to thank them for that. In between, we have attempted many crossing programs. I have not presented here. Day to day will change. There will be some challenges in ZRAC meetings. Journal Research and Extension has very advisory council meetings, and regional extension advisory council meetings. Will be getting many problems, and the farmers will be experiencing, and they will be just putting forth their problems. Based on that, we'll be formulating our research programs to tackle those issues from time to time. That is what is happening. Now we have very good program. Our lines, there are many lines also, advanced breeding lines in ASRP testing. Now I am hopeful that this flow of the material, it will continue in future also from our center. Thank you. Thank you.
Thank you, ma'am, for your talk, and the presentation was very nice.
Thank you, sir.
So now I request the participants to kindly interact with madam if you have any questions. In the chat box, there is one question from Suma Mughali from US Dharwad.
Sir?
Yeah, from Dharwad, US Dharwad.
What is that, Suma? I didn't see that.
Yeah, ma'am, can you, if you're here, otherwise I will read the question.
Ma'am, actually we're talking about pentaphyllate leaf variant, no ma'am. Does it
Yes.
have any economic importance, any trait of economic?
No. It doesn't have any exam. It is academic value only. It is of academic value only.
Okay, ma'am.
Okay.
Haji, good evening, madam.
Good evening, Narendra.
Congratulations, madam, for nice lecture. Nice to see you after a long time.
Thank you. I am also happy to see you all.
You have shared a good experience and you have developed a lot of variety. There is no option for that also, like Narayani, Dharani, Kalasti, Prasunna, Grishma. My question is what breeding methodology you have followed to develop these variety? Is it either progeny selection or bulk selection? What kind of breeding methods?
I mostly followed Narendra with only pedigree method, but it is not, as I say, that strictly pedigree, keeping pedigree record and all that.
Okay.
I will be selecting them in F2. I will be making selections. F3 again, F4, F5 also. As I said now, explained in between. This is, I think, we can say as modified pedigree method. And I kept on a record, actually, what are the number of selections that each time I am making, and whether that is going into field next time. Follow up that I have done. And after drying, I have verified the selections also. But in the
Okay.
lab, at the time of harvest, we'll be checking this filling and other traits. Otherwise, if the LLS, whether it is there or not, pod yield, pod number is there or not, like that, we'll be checking at the time of harvest, making selections. We'll be mostly concentrating on visual traits, but shelling out turn, all this SMK, again, I will be checking them at the time of shelling. And the time of shelling also, when you give after for sowing, you should be taking, you should be there personally. In the morning, you should go and give that material for shelling under your supervision only. That has to be done.
Because madam, selling outturn is a post harvest observation, but your all variety have good selling outturn.
That
More
is
than
what I am saying.
75%.
That is why I am saying that I am checking the material even afterwards, after drying,
Acha, after drying.
before sowing,
Oh,
before it goes for sowing.
okay.
In the lab, before actually, no, we'll be keeping the material drying and we'll be just keeping the material in drums or whatever the way we store. At the time of shelling, again, for next season, we'll be keeping the material for shelling. At that time, you have to sit with the material and see.
Okay.
Then only you can ensure.
Yes. And and in your breeding methodology, you have mentioned single plant progeny selection and uniform bulk also you have practiced
Yes.
simultaneously, eh?
No, in the same cross, there are many selections. There are many plant progenies, no?
Oh,
For
yes.
example, we have made in F3, for example, around 40 selections. Among these 40, we'll be selecting as a whole some UBs, or some we will be rejecting. From some progenies, we'll be selecting only single plants, like that. You have to modulate. You have to sit in the field and do. Some progenies we may have to select as a whole in F4. Some single plants only in some progenies, if they are not good, if they are not uniform.
Yes.
You rationally you have to do.
Yes.
You have to sit and do.
Madam, one question is that you have conducted a preliminary yield trial in F6 generation.
Yes.
There is a lot of chance of segregations
No.
that bulk.
In F6, no.
No.
Only if there is no segregation, only that kind of progenies only we will be promoting to yield trials.
Okay.
If there is segregation, we won't be promoting. We'll be selecting only single plants from them, from segregating progenies.
Okay.
Otherwise, only uniform progenies only will be promoting to F6.
F6, yes.
F6 and later on to yield trial.
Okay.
Segregating ones we won't promote.
Yes. Nice, madam. And what is the experience of JL24, madam, in your processing program? Is having high combining ability, or because that variety is widely used for
combining ability is good.
Eh?
Combining ability is good for JL24.
Good.
That is my observation.
Is good or poor?
No, it is not good.
Not good, but it is used in drought resistance breeding programs.
Yes, we have used because that is the line that is there, existing variety. So to replace that, we have used, but we got some material in that process.
And Dharani?
One line JL24 ICGV 86031 in insect resistance breeding program. That is TCS 647, but is not a variety. It is a good breeding line.
Okay. And combining ability of Dharani, madam? It is
good. It is good.
Because that can be used for transferring high selling outturn.
Yes, it is good, no doubt. Kadri 6 also is a very good combiner. Narayani is also a good combiner.
Yes. Okay. Thank you, madam. I am in Bikaner. Yes.
Okay, thank you, Narendra.
There is one question in the chat box, ma'am.
Sir, can you spell out that one, sir? I have not seen.
Yeah. Why recent varieties are not creating history like TMV2 and JL24? Question from US Dharwad again, from Yanagi sir.
Yanagi sir?
Yes.
Yes, madam. I asked this question not as an agronomist, as a farmer.
Okay, sir. I shall answer that question. TMV2, it no doubt is a variety. Now we see that variety, it is not a high yielder, no? Anyway,
Yeah, madam.
it is not a high yielder. When we bring in some genes, red route or other things, some undesirable, that way the seed and quality traits, they come in. That is the only disadvantage with our new varieties. But that has been overcome now in TCS 1694, for like the Dharani. Dharani did just one negative point is Dharani. After 6 months, that color changes. That is there. Otherwise, it is exact. It is no doubt. It is 100% good variety. It would have become a replacement to TMV. Definitely TMV2. Now also it is a replacement. Definitely. High shelling out and one more advantage with the varieties that they are clear Spanish bunch types. Early vigor, that is a very good trait that is there in those varieties, TMV2 or JL24. That is there in Narayani,
Okay.
in our variety. Okay.
But
These
again,
are some of the traits.
one more thing, what I observed in the central part of Karnataka, particularly with respect to TME2, that will raise both extremities of the climatic change, either low or high. That is the beautiful characteristics of actually TME2 elasticity in nature.
Elasticity,
We are observing high yield, madam, no doubt.
Rejuvenation capacity, they are there, but yield wise, they are much inferior.
Yeah.
And we have very good varieties, location specific. We have to use them. They may not be pan-Indian varieties suitable for all situations, but they have that advantage, yield advantage, resistance, other things also. They have really good varieties are good, no doubt. But they may not be pan-Indian varieties. No, to TMV2, okay, whatever the yield that we get, we harvest, but we won't complain. But when we new varieties, we ask them to grow. They farmers themselves, they express many negative points. They just when before adapting itself, they won't come that forward for new varieties.
Madam,
but now the situation is changing. Farmers are changing now. They realize the importance of new varieties and they are following the management options with the new varieties and they're happy now. No, I think there is no TMV2, at least in Andhra Pradesh. At least. Kadri 6 is there. Our varieties, Dharani, Narayani are there. And 1694, now it has taken upper hand.
Madam,
from Kadri also some varieties are doing well.
Madam, one more question. Actually, I am a member of Desi Cultivar Committee member of our Dharwad district. Is any variety or any cultivar which is to be called as a Desi in groundnut?
Desi, na?
Desi, madam.
Actually, no, variety groundnut itself is an introduced crop.
Okay.
We can Desi we can't say, I think. If it is our country's the place of origin, then we can say.
Okay.
Like that.
In
Dr. Muralidharan is there.
Yeah, madam.
Dr. Muralidharan, sir.
Yes, madam. I'll give comments after this piece. Okay. Let them complete. The questions let them complete. Okay.
Okay, sir.
I have very high regard for Tirupati varieties. I will tell you that the reason why that varieties are very good. I'll tell you. Okay. We were talking about TME2, na? I'll tell you. Narrajan, shall I talk?
Okay, so now
yes, sir. If any other questions are there from the audience, sir, we can. There is one question from Shrikant, sir. Shrikant, sir.
Yes, sir.
Yeah, you can ask.
Yes, sir. Thank you, madam. Very good lecture. And so actually we have interacted many times in workshop also. I am from Raigad Center of IGK with Chhattisgarh. Generally, ma'am, that in your that variety, right from Tirupati one to latest one, that duration and yield, that just I want to clarify. Actually, that duration in Kharif it is higher and summer it is less. Then yield also having difference. Just it is double in summer. So in summer season, getting higher yield. So whether it is genotypic difference or only environmental difference.
It is environmental difference also. Environmental difference only. Actually,
Yes, ma'am.
in our place, it is not summer. It is Rabi. We take up sowing in November, December. Our crop comes to harvest by March.
Yes.
That is the situation. We guessed almost, actually, our farmers reap more than our research yields, actually, research station yields.
So
Almost
same
more
thing
than double also
repeated in our
sometimes.
Chhattisgarh. Yes, ma'am. Same thing
Okay.
repeated in our Chhattisgarh also. In our Chhattisgarh also, that yield goes high up to 7 ton in summer season.
Hmm.
So that is unbelievable in our some part like Ambikapur, Sarguja, northern hill parts, and it was very higher yield. So
Yes,
that,
it is
but no one is
possible during
believing.
Rabi season with good management.
Yes, ma'am. Good management.
Yes.
Good management and low incidence of diseases and insect pest also.
Season also. Season
Yes,
climate.
season also. Assure, you have assure irrigation.
Yes.
Yes, sir. We have assure irrigation in summer, not
Yes.
for more area, but whatever in area they are growing, having assure irrigation and getting very good summer yield as compared to Kharif, sir.
Yes.
That is possible. There is no doubt about it.
Yes, sir. Yes. Thank you, madam.
Any other questions? Regarding the drought traits, that in our last RAC also, Institute RAC was there last on 20th and 21st.
Yes.
So they also suggested to go for the surrogate traits, identifying the surrogate traits for drought, because you have worked for the almost your entire tenure on drought tolerance.
Yes. Yes.
So, and I see that a lot of your varieties released having higher shelling percent.
Yes.
So our Ajay sir, who is heading Anantapur station, also believes that the shelling percentage is one of the surrogate trait for drought tolerance. So what is your
It is not
opinion?
a surrogate trait, shelling out, and it is a right yield trait that should be there under right rainfed situation. If the genotype maintains shelling out, and it is a very good genotype, no doubt about it.
Okay.
It is like we can't say that is the surrogate trait for drought resistance. It is a yield trait that if it is maintained under drought situation, definitely that is a very good genotype. That is how we have identified Dharani TCS 1043 during Kharif 2009. That is why I have put up that case study also, actually, as a case study. It was it recorded 71% under such a worst situation. That is very important. Shelling out, and no doubt it is important. But yield also we have to look into. Only shelling, no. Along with yield, it should go.
What should be the yield penalty, madam, for drought tolerance? If any variety has a drought
Yield
tolerance.
penalty, we can't say that. Actually, in TMV2, if it is more than 15%, if it is giving more than 15 to 20% over the existing variety, along with the drought resistance, there should not be any penalty as it is. There should not be any penalty. It should have higher yield for the existing variety along with drought resistance. That is what happened in Dharani. In rain-fed as well as in favorable season, you have seen Kharif 2007, it has recorded very high yield. That was more than 33%, I think, for the Narayani and K134. We should look into both yield as well as shelling out and resistance. We should consider all these together. In such a way, we have to choose our parents also.
And while compiling the compendium for varieties released in the last 5 to 10 years that we were last year we released in the AGM. So I have one question. In Tirupati varieties, I see water use efficiency and drought tolerance words are used for some of the varieties. So can you
what is it? Water use efficiency, it is frequently used.
Some of the varieties have water use efficiency special traits. But
Hmm.
one or two varieties have drought tolerance also, like
Yes, I
mid
can
or
say
late
that.
season drought. So can you like differentiate
Actually,
between these two?
the water use efficiency doesn't mean that it has drought tolerance.
Okay.
Dharani, it has drought tolerance. It has to perform well under drought situation if the dry spell is more than 25 days, 30 days. That is again a trait that we have to, empirical trait only, that we have to evaluate and decide. If these water efficiency traits, SCMR, all these, if they are good, it doesn't mean that it is a drought resistant and we can promote them for rainfed situation. Some of the traits, they have high water efficiency. For example, 1073, it has high water efficiency, but we are not promoting that for rainfed situation because of its pod and kernel features. We have to look into that aspect also. It has slightly bigger pods. And when it is put in rainfed situation, we are not getting shelling out in there, in those genotypes. Via media, we have to strictly, if we stick on to those traits only, you have to combine both empirical and trait-based, whatever you call. We have to think of in both the ways also. We have to evaluate them and rainfed situation. And you have this information for these surrogate traits also and decide. It is not that we have to, the water is efficiency because actually even insect resistant breeding lines, insect resistant breeding, they had higher water efficiency. Because of the thick leaves and all that, SLA high, low SLA, they will be having thick leaves and dark green foliage. All these traits, they confer directly this water efficiency. Physically now we can just, instead of going for SPAD, we can physically select now. That considering all these things, if we are so experienced and if you closely follow the material. Okay, we need not have to have this SPAD chlorophyll meter for each and every plant. We not have to go to see what is SPAD and all that. Rosiness wise, we can just judge.
Thank you. Thank you.
Thank you.
Now, Mridhar sir, open.
Actually,
Sir.
Vasanthi, thanks, madam. Actually, we have seen that this one groundnut.
Sir, your voice is not audible, sir.
Oh, wait a minute. Is it audible now?
Yes, sir.
Okay. Actually, I'm very much thankful for your detailed lecture, and we could see that journey of groundnut in Andhra Pradesh through you. Actually, to our that breeders, you might have seen the varieties released from the Tirupati Center exclusively for rainfed situation. You might have seen that medium parts of 2 kernel types, because that is very important. As that is, if you see that the automatic shelling will be also increased, will get increased, because that ovary is that pod is an ovary, it's a maternal tissue that will develop as the plant develops. But whereas inside the kernel, they are reproductive parts and due to fertilization. So after 85 days, if there is any drought happened in that, mostly happens in rainfed crop, so that pod only will be developing, that kernel will not develop, so that rattling will be there and the shelling percentage is reduced. But as Dr. Vasanthi, she has selected only that small to medium parts, 2 kernel types mostly. That is the reason we get that yield because TME2 is like that only. 85 days, you can see that full matured kernels in TME2, but it's highly susceptible to diseases. We have very good varieties from Andhra Pradesh and Tamil Nadu, also from Kadiri, that to replace this one, also from the Dharwad also, to replace TME2. Because whenever the breeders go for exclusively for rainfed situation, they should not go for bold parts, because that shelling pattern will not be there, full kernel formation will not be there. So that if there is irrigation facilities available, they can go for medium to bold parts. But for irrigated and the summer crop, that is okay. the duration also not in a question. Okay, this is the main secret in our rain-fed this one breeders selecting indirectly selecting for medium parts to kernel seeds, and this is very important with a slight beak. And from a lecture, we could see that Bima, I told you in my lecture also, the mutation is not only for direct releasing for mutants, but it's mutant into mutant for pedigree selection also. We can use it. So that same thing is used. And that in 2003, she has written a paper on this pentafoliate leaf. She has given that is it's a recessive character. There is one variety, Pengua 1 is there, I suppose, Pengua 1. It is a natural mutant for this one. Pentaleaf and pentafoliate, and this other one 15 16 that there is developed from TME10. TME10 is a natural mutant in 1970. It was identified in germplasm, agent germplasm in Tindivanam. It's having a variegated star. In Vernacular, Namakotai, red and white, Namakotai, Namam. And there is a Nambigore also there, Arachis Nambigore, that is also having the same thing like that. But dormancy more than 2 months, very dark green foliage, and tolerance to many diseases also. So I think across the country, she has alone used that for the development of one variety. Thanks for her. And thanks to her, because the TME10 we don't have as on date in Tamil Nadu, but it's a very good variety. Having
We don't have
Kemal
that, sir, but
Concolate.
TCZ 1518 is being maintained by us.
Yes. Thank you very much for your lecture. And one more in lighter vein. I think you are from your home. You are telling you are this one giving lecture, na?
Yes, sir.
I think your wall clock is not functioning.
Yes, sir. That is not functioning. Okay, yes, sir.
This is breeder observation. Okay.
Okay, sir. Okay, sir. Thank you, sir.
Okay. Thank you very much.
Thank you, sir. Thank you, sir.
Okay.
Thank you, sir, for your interaction and session. Appreciation, whatever is. You're a guru for us, guiding
Thank you.
post. Like Dr. Nigam and Dr. P. Reddy, you were there with us, sir, from the
Thank you,
very
madam.
early stage. So thank you, sir.
I was waiting for your lecture.
I request other participants if any questions are there. Then, okay, so I don't think any other questions are there. So with that, I thank Vasanthi ma'am for because she was traveling and she agreed to present after several requests, and she made and after that madam was in regular touch with me over the phone, over the WhatsApp, that she is eager to present, and she prepared and shared the slides with me. Thank you ma'am for that. And as director sir is away, he's in ICAR side, and he's with the DG. He has some presentation today, so he couldn't attend. So he expressed thanks to madam for accepting our invitation and presenting today. And as I see, many of still the maximum strength reached was around 45 today. Thank you, ma'am. And we are very much benefited from your presentation. And if you agree that, can I share some of the slides to our participants in?
Definitely, you can share entire
Yeah.
PPT.
Thank you, ma'am. Thank you. So I will share
Thank
that.
you.
Yeah, we've already recorded this one, and soon we'll be putting up on our YouTube channel also. So thank you, ma'am. Thank you, and we look forward for interacting with you again sometime. And this year's AGM is in Udaipur, so we already sent a request email, circulated email. Not in the message I have circulated. Email, the letters will be sent soon. Thank you, ma'am.
Thank you, Nataraj, and I thank Dr. Bera and others at PGR for giving me this opportunity. And this is also a good opportunity for me to interact with all the young breeders now, upcoming breeders, so that I can share whatever I have learned during my journey with groundnut. That is such a actually golden opportunity, I think, given to me. In spending 30 years, it is not a easy thing in state agriculture universities. I think I have to thank your university also for giving me this opportunity. Thank you.
Thank you, ma'am.
Thank you, ma'am. Thank you. So
Thank
with
you.
that, yeah, with that, I am signing out. Thank you. Thank you, sir.
Thank you, Natraj.
Thank
I
you.
have given you trouble in sharing that.
No, no, no.
Okay,
Thank
thank
you.
you.
Thank you, ma'am. Have a
Thank
good
you.
day. Have a good day.
Okay, thank you.